Hello, Harry Brook here with Flagstaff County. And today we're talking on our podcast with Dr. Michael Harding, a pathologist with Alberta Agriculture and Forestry. And today we're going to talk about some of the recurring diseases that are showing up in fields and what we can look for in this coming year.
You're listening to the official podcast of Flagstaff County.
So, Mike, how about the Fusarium survey that was done last year? I know that it was taken off the Pest Act, but there's still a lot of concern over it.
Yeah, rightfully so. Fusarium isn't your run-of-the-mill crop pest. It's really difficult to manage, and you have to really take a multifaceted integrated pest management approach to be able to kind of defeat that one. And so, yeah, we did a survey in 2020, about 450 fields across the province, which represents about 1% of the acres in the province. And we don't have those results yet, but what we do have for 2020 is the results from the Canadian Grain Commission's harvest sample program.
Oh yeah, what did it show?
So the last time we had a really serious Fusarium head blight issue in the province was 2016, which you may remember was a pretty wet year, almost from top to bottom in the province. It's a really good year for a plant pathologist to see lots of diseases. And then it got quite dry, relatively speaking, in 2017 and 18. And then in 2019, the FDK levels were back up around 25%, 25 to 30% of samples with FDKs.
And how severe was it in those samples?
The severity in Alberta generally ranges from about 0.5% to 1%. So we don't have the high severities that we see in some other places. But we, prior to 2012, we never had more than about 5% of samples with FDKs in them. And then in years like 2016 and 2019, we've got 30+% of samples. So we're definitely seeing more. Now, in 2019, it was really quite surprising to me that there was almost 30% of samples with FDKs because where I am in southern Alberta, it was quite dry.
Yeah.
I would not have predicted a significant amount of FDKs. And so I took the Canadian Grain Commission data, and they also, you can get the data for the whole province.
Yeah.
Or you can get it split up by crop district. And so when I looked at the individual crop districts, I saw that the southern Alberta districts, crop districts 1, 2, and 3, had less than 10% of samples with FDKs in them, but it was crop districts 4, 5, 6, and 7. So in the central and the north that had those high levels of, up over 30% of samples with FDKs in them. So that's the very first time we've ever seen anything like that in Alberta, where the central and the north have more FDKs than we did in the south.
I was just wondering, is that an indication then that really we have the inoculum is all through the province? We just have to have the right weather conditions for it to show up?
I think partially that's right, Harry. So we have more places in central and northern Alberta where it's become established. That's not to say it's established everywhere. It's not.
Yeah.
But we have more areas in central and northern Alberta where it is established. And so then in a year like 2019, where you get that conducive weather in the north, but not in the south, then for the first time, we're seeing that split where we've got the higher levels in central and northern Alberta, whereas usually southern Alberta has the highest levels.
That's where it started showing up 10, 15 years ago, wasn't it?
Yeah, so the situation is really changing in Alberta. And so for that reason, I think if I was a producer and I really had never seen a lot of Fusarium head blight damage or Fusarium head blight issues on my farm, I wouldn't assume it's going to stay that way forever. So I regardless of where you're standing in the province now, this is something we all need to be watching for. And then in areas where we know that it exists, we have to take special care to manage this disease because it's challenging. It's really challenging to put a lid on it.
How long can the spores last in the soil? Or like it's in the crop residue from the previous year's wheat, right?
Yeah.
So
it doesn't really produce a long-lived resting spore or long-lived resting structure that's going to survive in the soil for a long time. So it really is only just present in the crop residue. And so, crop rotation can certainly help to manage this disease. But we do have a lot of cereal acres in Alberta, and it's not just wheat that it can colonize. It can colonize barley and oat and rye and corn. So any of the cereal crops. And so it's present in enough fields that you're, probably almost always close enough to just about throw a rock and hit, some crop residue that may have fusarium on it.
Yeah, Central Alberta's crop rotation tends to be very limited to canola wheat.
Yeah, so in a situation like that, that puts one at a pretty high risk for that pest, for Fusarium graminearum, to become established and start causing some downgrading issues for cereal producers that are growing barley or wheat.
Could you possibly go over like the symptoms you'd see in the head, since that's usually when you first see it, right?
Yeah, the best time to watch for the symptoms is kind of at the early milk, soft dough stage, when the head is still green. And then any of the florets or spikelets that are infected will prematurely ripen. And so you'll see a green head with some bleached florets or spikelets. And so that's the best time to scout for it. It's not easy to see the symptoms. And so, you're certainly not going to see them driving by on the road.
No.
And even when you walk right past a head that has Fusarium head blight, it's hard to see. You have to sometimes even almost look under a microscope and peel the gloom back to see evidence of the disease.
Oh, yeah.
So it's not easy to see, but if you do scout during that late milk, early dough timing, just before the head starts to turn, sometimes you'll see those prematurely ripening heads. And if you see any kind of salmon or pink or orange colored growth on the head, that sometimes will be an indication that you have Fusarium there. You don't always see that, but
could it be something else if you saw that?
If you're seeing a salmony pink colored fuzz growing out from under a gloom, that's probably 100%
indicative.
going to be Fusarium.
Okay.
But sometimes plants can change colors for other reasons too. And so if the plant is just looking yellow, or
Purple,
kind of got a funny color to it, and it's just all over the whole head, that's maybe not Fusarium.
Okay. Well, let's talk about some of the other diseases out there. What about pea diseases? Because there's a lot of pulses out there, but we're also hearing more talk about Aphanomyces, and there's always Ascochyta mycosphaerella that are out. And these don't seem to be going away.
Yeah, you just nailed the two most prevalent diseases in peas, root rots. And most of our root rots in Alberta are caused by Fusarium, but Aphanomyces euteiches is becoming more and more widespread each year. It, in conjunction with Fusarium, can be really, really destructive. In some fields, for sure, is the biggest problem that a grower would have producing peas. So yeah, Aphanomyces can be really destructive. We don't have the number of fields with Aphanomyces yet. That work is still in progress. A lot of the lab work was kind of suspended during the summer because of COVID.
Right.
And that's why we're kind of running behind on some of this. In the past, we've seen more and more fields reporting positive for Aphanomyces. So Fusarium's still more common and still probably doing, the lion's share of the root rotting in peas.
Yeah.
But where Aphanomyces is present, it gets really severe.
So would they be complex together, like you'd find if you're going to find Fusarium, or if you find Aphanomyces, usually you find Fusarium as well?
Yeah, probably not a complex in the truest sense of the way that term sometimes is used, but they can co-occur for sure.
Because they occur in the same conditions, right? Which is lots of water in the soil.
Yeah. So that's a real troubling one. And again, the reason Aphanomyces is such a concern is that we just don't have a lot of great management options. And so, this holistic integrated pest management approach to dealing with it is the way we have to go. We don't have great dominant resistance gene that we can just throw at it to solve it.
Chemical.
Don't really have any chemical that is going to solve this problem for us. And so really, unfortunately, for fields that have high levels of Aphanomyces, the best recommendation is to just don't grow peas or lentils there for a while. And when I say a while, I mean like probably 6+ years. Not every field that has a Phantomyces has to go out of peas for, an extended long rotation like that. But that's why you need to scout and keep records.
Yeah.
And so if you see that root rot level sort of ratcheting up in one of your fields, and this usually happens in fields that poorly drained or have lots of compaction issues.
Lots of low areas.
Yeah. And
Water
so
puddling.
these are fields that aren't great for peas anyway. And then when you add the root rot on top of it, it just becomes kind of a no-win situation. So kind of avoiding those fields where you can. And unfortunately, both Fusarium, a couple of the species of Fusarium and a Phantomyces do produce a long-lived resting spore or resting structure that can survive for, say, 8 to 10 years in the soil. And that's why fields that have had peas or lentils in rotation for, say, 20 or 25 years, some all of a sudden will start to just have these, catastrophic root rot issues. And it's just because the levels ratchet up over time. Even if you have a good 4 to 5 year break, they just survive long enough that you slowly ratchet up. And then all of a sudden you cross that threshold and you got a big problem.
I noticed because when I talk to producers, sometimes they'll say, I put peas into this field for the first time, had a fabulous crop, and it's never produced as well with peas ever again. I'm wondering if it's got something to do with slight or slow buildup of long-term disease spores.
Yeah, that can definitely be one of the factors that leads to that kind of an observation. Yeah.
And how can you tell the difference between the Fusarium root rot and the Phantomyces?
That's got to be a lab test.
Okay. I thought with Fusarium, if you cut the root near the bottom and if it's red, it's got Fusarium in it.
So I've cut hundreds of roots open that have Fusarium, and I've never seen that. So if you get lucky and see it, send me a picture, because those pictures are really cool. But that's not a diagnostic symptom that you can really hang your hat on. So if you can tell if it has root rot. Yeah. And so if you see a lot of sort of honey brown discoloration of the whole root system, and root pruning, meaning that like just a measly root system, that's usually more indicative of Phantomyces. If you see discrete sort of black colored lesions, especially at the crown, near the crown of the plant, that's usually a little bit more likely to be Fusarium. But those symptoms run together a lot of the time. And if you want to know if a Phantomyces is there, you need to send it to a lab test. And I think both 2020 and BioVision perform that test. It's commercially available. I would just contact your favorite seed lab or diagnostic testing lab and have that test done if you want to know for sure.
Well, do they also do Ascochyta and Mycosferella testing? We're moving on to a different disease.
Sure, yeah, no, that's fine. I think they do that on seed, but I don't know if they'll do that on plants. Now, Mycosferella ascochyta is pretty easy to recognize. It just causes flecks or lesions on the leaves and stems. Yeah. And, the majority of the time that I'm in a pea field and I see those lesions, those dots or spots or flecks, lesions on the leaves and pods and stems, that's Ascochyta mycosferella complex. Right. And it's the most common pea disease in Alberta. We see that in 100% of fields. Yeah, at varying levels. Some fields have very little, some have lots, but we see it everywhere we look.
So the Ascochyta mycosferella, would you say it's a major factor in peas falling down? Because it seems you get those lesions in almost all the pea fields I was in this summer. I noticed lots of lesions right at the base of those plants. Black lesions, either not girdling it, but certainly they were there. And this was fairly early on, and it was still flowering.
That's the theory. There might be a published paper out there somewhere that I haven't seen that proves that that's the case, but I don't have any scientific evidence or proof. But it stands to reason, and it seems like a logical hypothesis, that if you've got a pest or a pathogen that's chewing away at the stem and making it, a little less structurally sound, that then all you need is a bit of wind and that canopy can start to tip over. So I can't imagine that Ascochyta mycosferella, especially the foot rot that Ascochyta causes, I can't imagine that does any favors with respect to standability. So I would agree with you that it's a problem. And it's the reason why, if I'm in a pea field and I see a lot of ascochyta foot rot down low in the canopy and the grower's thinking about a fungicide application, I say to them, don't scrimp on the water volume, because you want to push that fungicide down as low in the canopy as you can.
Yeah, it's so difficult, isn't it, to get it down that low?
Yeah, and there's a fine line between waiting, because if you apply too early, those products only have a, say 7 to 14, maybe if you're lucky, 20-day window of protection.
Yeah.
And so you don't want to put them on too early, because then the disease just comes back and causes the problems. But you wait too long, and now the canopy's so rank that you just can't get your active ingredient down to where it's needed. So that's a tough one. You got to try to find the sweet spot if you can.
And I know that producers, they absolutely hate the idea of 20 gallons to the acre, just because it means you don't do it in one pass.
I feel their pain, and if there was a better recommendation, I'd give it.
Yeah.
But if you want to get the active down low, you got to have water.
So do you have any results this year on any of the canola diseases?
Yeah, for sure we do. So we cooperate with Dr. Stephen Strelkoff at the University of Alberta on the provincial clubroot survey. And then we lead the blackleg sclerotinia verticillium survey. Yeah. So we've got all these results.
Did you find much in the way of verticillium this year?
We haven't seen it in our survey. And this year we had 4 fields that had some really suspicious symptoms that looked like it was going to be verticillium. And we thought maybe we'd found it. And so we had them tested, but they came back negative.
Oh.
3 of the 4 were positive for blackleg. And the 4th one, we don't know what it was, but it did not test positive for verticillium. So I'm not suggesting verticillium is not present in Alberta.
Yeah.
I'm just saying it's at low enough levels or cryptic enough levels that we're not picking it up in our surveys.
When you mentioned blackleg, and I was thinking how when I was doing the canola surveys, there's lots of it in the fields. Yeah. You find it everywhere. But why are some fields more susceptible than others? Is it like the genetic mix of resistance they've got? Because I remember about 3 years ago hearing a producer phoned me and said he lost 25% of his yield due to blackleg. Yeah. How much of that do you think would be linked to just the varieties and the mix of genetic resistance?
That's a really important question. And we generally, across the province, are seeing a decline in the prevalence and incidence of blackleg. Over the last few years, the numbers have been dropping a little bit every year. And again, it's like you said, if you looked hard enough in every field, you probably could find blackleg just about everywhere. Yeah. In our survey, we don't look at every plant in the whole field. We walk a pattern and we look at, 10 plants at this spot and 10 plants at this spot, et cetera. And in doing that, the numbers have been dropping a bit. But what we have noticed when we look at our survey data is that we can put the survey locations onto a map, and then we can tell the computer to look for spots that have higher than average severity, and then look out next to them and see if there are other spots around them that have higher than average severity. And if you see multiple spots with higher than average, then call that a hot spot.
Yeah.
So we do this hotspot analysis, and we found that there's kind of a strip kind of in the center of the province that's more prone to these hotspots. And we're in the process of doing some work to try and really track down what is causing this. Why do you have, say, 5 fields within 100 miles of each other, and 3 of them have, serious levels of blackleg, and 3 of them you almost can't find it? Why is that? And there's a number of factors why that could be, including what you said. So variety selection and, the match of the resistance genetics to the races of the blackleg pathogen. Right. That's going to be an issue. The crop rotation is going to play into that, and the weather is going to have something to say about it. And so there's a number of factors that could be driving where these hotspots are showing up, and we're hoping to do a little bit of work here over the next few years to sort out which, what's the main driver or drivers of these sort of blackleg fields or these canola fields that get these severe levels of blackleg.
Because I noticed it in the survey, there'd be some fields and you could find traces of blackleg in almost every stem they were pulling. Not that it was severe enough it affected yield, it seemed. And there was others where, you'd find maybe 1 out of 20 or 30 stems that had a little bit of blackleg and they were clean. And I mean, considering that it's a 2-year rotation for a lot of guys with canola, you'd think that the infestation or the infected rate would be a lot higher.
Yeah, like you pointed out, it depends on which races of blackleg are there, and if the resistant genetics are still effective against the races in that field, then there's no issue. But if that once that population kind of shifts to a virulent race, then you start to see the issues popping up. But
we're seeing it going the other way, right?
Well, we're seeing the overall levels dropping. That doesn't mean we're seeing fewer fields that have, these dangerous levels.
Yeah.
Does that make sense?
Yeah, it does.
So
there's still lots of fields showing signs.
Yeah. The overall prevalence and incidence is dropping, but we're still seeing hotspots.
Yeah, where it's really bad.
That's what we're going to try and figure out is what's the drivers for these hotspots.
Okay.
And we're going to be doing some really detailed sort of high intensity surveys in canola fields in these areas that are showing these hotspots. And we're going to need help from people like Ag Fieldman, Harry. So if you're
I think there'll be some bodies out there for sure. I'll help you.
If you're interested in helping us sort this out, that would be great.
What about sclerotinia? It's always around, but I don't think it seemed to be that bad this year, at least not in our area.
Yeah, I was a little surprised because we certainly had lots of moisture that could have sort of got those sclerotia bodies germinating and forming apothecia and releasing the spores. But maybe in some areas it was so much moisture that they drowned the sclerotia out and they couldn't form the apothecia. And also it did get quite dry mid-season during the bloom period in some areas. And as a result, we had quite low levels of sclerotinia in our survey. We didn't see very much of it at all.
Yeah, I was out in the fields. I didn't see much of it either. And I was surprised because we were pretty wet right through until the middle of July. So that would have been well into flowering. There should have been a lot of infection kicking around. But
sometimes it can be too wet.
Yeah.
So if the soil is too waterlogged, then you, instead of germinating, those sclerotia can drown.
Okay.
So depending on where you were in Alberta, one of the biggest issues that I saw in pea fields in central Alberta and parts of northern Alberta was drowned out areas where the peas just died because there was water standing for so long. So we sometimes think more is better when it comes to moisture, but
there's a limit, isn't there?
Yeah, anybody who can't swim knows that you can have too much water.
Yeah. And the club roots, because you worked with Dr. Strelkov on that.
Yes.
Just continues to show up in more places, eh?
That's the story. Yeah, for sure.
Yeah.
About 10% of the fields that we evaluated were positive. That's out of about 600. And we had 3 counties reporting for the very first time. So Smoky River and Grand Prairie in the Peace region, and then Wheatland in the south. So it just continues to sort of push on that growing fringe and showing up in new counties. So this is another disease that, like Fusarium head blight, if you live in an area where you've never seen clubroot before, I wouldn't assume that it's going to stay that way forever.
Yeah.
And as a result, you need to be scouting for it.
Yeah, you don't want to be surprised and lose half your crop, right?
No, and there really isn't anywhere in the province now that you should be growing susceptible varieties. We're everywhere that we grow canola now is close enough to this growing front of infestation that
it's potentially there already.
Yeah, we should be deploying that genetic resistance to try to keep those spore loads low, and then don't grow canola. Don't use that resistance more than once every 3 or 4 years. So
Okay.
crop rotation is going to be important, because when the clubroot pathogen produces more resting spore, those galls get filled up with resting spores at the end of the season. And those resting spores aren't just one pathotype. There's multiple pathotypes that get produced in those galls, and so the resistance breaking pathotype is already there.
Yeah.
If you use the resistance too often, you just select for it, and then all of a sudden you got a dead patch in your clubroot resistant canola. So yeah, using the resistance in advance of to keep the spore loads low, and then a good rotation, a long diverse rotation to help so that if it does get introduced, enough resting spores can die before you bring the next canola crop or next host crop in.
Do you keep track of how many areas have reported, I guess, breakdown of the first resistance genes?
Yeah, Dr. Strelkoff is working on that. And that's a challenging one, because we don't have rapid tests to determine what pathotype it is.
Mm-hmm.
They actually have to do a biological assay on a differential set of canola and vegetable crops.
Right.
And it takes 6 to 8 weeks to do that in a greenhouse, and you have to replicate it because there's some biological variation within that system that you have to account for and do statistics, and it's a lot of work. And so they are doing that and trying to keep us informed of how things are changing, but we're always about a year behind with the results because it takes so long to generate them. And
would having to use the biological route.
Yeah. So the prevention of resistance erosion with good stewardship and crop rotation is really the way.
So let me throw you a curveball.
Nice.
So tell me, have you heard much about any diseases in flax?
I visit a flax field or two just every year or every other year.
Yeah.
And with the exception of Fusarium crown rot and some root rot issues, they tend to be pretty trouble-free. Like, we don't have the pasmo issues that they have in Saskatchewan and Manitoba. So most of the flax fields that I see, we don't produce flax in a real intensive way that we do wheat and canola. Yeah. And so, they tend to be pretty trouble-free.
I knew I just threw it in there because I was thinking how, with the price of flax being so high this year, that I think there might be an encouragement for more acres of flax to show up. And there's some impressive yields last year from flax. So I was just wondering if we might even see a little more flax out there, and then with that, a few more diseases.
Yeah, I admit I don't get into very many flax fields, and so there may be some issues there. We don't survey them. We don't do disease surveys in flax, so there may be some things I'm not accounting for. But based on what I've seen in the few that I visit, they tend to be relatively trouble-free in comparison with things like peas and wheat and canola that we grow a little bit more intensively here.
Yeah, build it and they will come.
Yeah,
talking about the diseases.
Exactly. I'd like to mention something that guys should keep an eye out for in 2021.
Yeah.
We talked about some of the diseases we already know about and that are here quite routinely, but we've got a bacterial disease in wheat and barley that's making a bit of a, I guess, it's emerging.
Okay.
And so it's not like it's never been here before. It's not a new thing, but it's rarely an issue. Usually we see it and it's just a curiosity. Oh, look, there's a little bit of bacterial leaf spot or bacterial leaf streak in wheat. And then we just move on because it's not an economic problem, but that's changing.
Oh, yeah.
And so in 2018 and 19, we started seeing an increasing number of fields that had symptoms, and even starting to hear anecdotally of 2 or 3 or 4 fields that had economic levels, like losing up to 20% of their yield.
Wow, that's getting serious.
Due to bacterial leaf streak. And so we've started looking at this and doing a little bit of work on it. And the key piece where we're at right now is that We just want people to be aware of it.
Mm-hmm.
That, if you start to see lesions that appear like a long streak running parallel with the vein of the wheat or the barley plant, don't assume that that's a tan spot lesion or, in barley, a net blotch
Septoria, yeah.
or septoria. It could be a bacterial pathogen. And as a result of that, the preventative fungicides that we use to control tan spot and septoria and stripe rust, they're not going to work.
No, it's just a waste of money.
Yeah. So keeping your eye out and then getting a good diagnostic is really important.
Mm-hmm.
And if you think you see, so lots of times for anyone that's seen stripe rust,
yeah,
lots of times those lesions look just like stripe rust that's had those orange Uridina spores washed off.
Okay.
So it's kind of a long streak or a strip that runs parallel. That's the symptom that we're seeing. Now, there's other bacterial pathogens of wheat that will cause spots. And in oat, we see halo blight,
Mm-hmm.
which is more of a lesion or a spot. But in wheat and barley, what we're seeing is this leaf streak. The bacteria can also splash up onto the head and cause a gloom blotch.
Yeah.
And then they become seed transmitted. So this is a really important reason to watch for it. If you have bacterial leaf streak and it turns into a gloom blotch and you harvest that grain and then use that next year for seed, you're going to have a real bacterial leaf streak issue in that crop. Because
And seed treatments won't touch it.
We don't have any bactericidal seed treatments registered, I think, for any of our crops. And so, and it is really, a lot of bacterial pathogens can be really effectively seed-borne or seed-transmitted.
Mm-hmm.
And bacterial leaf streak can be both. It can either just be passively associated and be transmitted, or it can be internal and be seed-borne. So if you see a streak-like symptom in your wheat or barley and you don't know for sure what it is, keep your eye out for it and keep some records. And because if it is bacterial leaf streak, you don't want to use the grain from that field as a source of seed for the next year.
Can you use a lab? Like, does the Alberta Agriculture and Forestry lab, will they do some pathology testing for that?
So to know if it's a bacterial pathogen is a
Yeah.
pretty easy test. It's just a bacterial streaming. You could probably get that done at any lab that has a microscope
Okay.
that does testing. So the plant health lab could do it. My lab could do it. It's a good idea. Probably any trusted agronomist or plant pathologist could do it. And if people need directions or instructions on how to do it, I'm happy to provide that.
Okay.
But the key is to be watching for it. And if you see something suspicious, then, start reaching out to find out who can test it and figure it out.
Nip it in the bud before it nips you in the butt, you might say.
Yeah, it's only going to get worse if it goes undetected.
Right. Okay. Well, thank you very much, Mike. I really appreciate this.
You're welcome. It's my favorite thing to talk about. It's plant diseases. So I've enjoyed it.